Was it HPV?
No, this was done back in the early 1900s, and it was actually the tobacco mosaic virus. It was actually something that they studied in a lab, and that's how they figured out that a lot of cancers were actually either from a virus-like. Mm-hmm. Yeah.
So interferons are not aren't non-specific, are they?
Yes, they're non-specific. This would be, in other words, it doesn't matter what virus you might have gotten infected with, it's going to act the same every time.
Okay.
Yeah. Now, our complement system, not like, oh, I love the sweater today. Okay, note the spelling. C-O-M-P-L-E, not L-I, okay? Globulins, ooh, do y'all remember that term? What were globulins?
That's what they.
Go ahead. Okay, okay, we're right, we're on the right pathway, okay? A glob, all right, do you guys remember how they were in the blood, okay? And it was like a protein, all right? And they're used in many ways, all right? They're used in a lot of different ways. This time, that glob contributes to my non-specific and adaptive immunity, meaning the way that my body is going to act every time under non-specific resistance. The methods of action that it helps with: inflammation, immune clearance, phagocytosis, and cytolysis. It has some pathways. Your textbook is going to get in a little more detail. It's interesting to read. The classic pathway requires an antibody. Uh-oh. What? Isn't that more specific? Yeah. But just hold on, because we're getting ready to get too specific, okay? It requires an antibody that makes it part of adaptive immunity. Meaning, when we get too specific immunity, we're still going to see classic symptoms. We're going to see pain, swelling, fever. A lot. Okay, not every time, but a lot. There is an alternative pathway for the complement. This one doesn't make it specific and keeps it non-specific, meaning inflammation, immune clearance, phagocytosis, cytolysis. Go in, go in, get to the pathogen, try to kill it, and get rid of it. Which means they have to go in, they have to eat it, they have to break it up, they have to go through all those steps. But just hold on, because we're getting ready to get too specific where we got to talk about antibodies. Okay? Now, the other thing about natural killer cells, making them non-specific, okay, is the way that they act to kill something. It's really cool what they do, okay? If a natural killer cell encounters something, here's what it'll do. If this is my pathogen, here's my natural killer cell, the natural killer cell will attach. one of the things that it'll do, it will perforate the pathogen because it releases perforants. It will perforate. What does it mean to perforate? Poke holes in it. If it does that, one of the things that can happen, water can rush into the cell, but at the same time, the natural killer cell releases granzymes, which are basically a enzyme that'll enter into the target cell and help to kill enzymes inside that target cell. So, natural killer cells will encounter something, perforins get released, that breaks holes in the plasma membrane, that allows water to rush in. Natural killer cell is also injecting granzymes. That pathogen goes, whoosh, it bursts. And it's now dead. What was our first antibiotic?
Penicillin?
Penicillin. How did penicillin kill a bacteria? Penicillin is not specific. Penicillin was used against any type of bacteria, and it was used against any type of bacteria because of its mode of action. What it did was it created holes in the walls of the bacteria. Water from the body rushed in and blew them up, just and killed the bacteria. That was it. That's pretty cool. Now, if you get sick and you're given an antibiotic, because we have created such wonderful bacteria now, okay, because we have created them, we've had to get more inventive on how to treat them. So now, when you go and you're sick, you get an antibiotic, you go fill it, might cost you $800 for 2 pills. Why? They've had to engineer that antibiotic to the point that it can actually enter that particular bacteria. They've gotten more specific, okay? Enter into the bacteria, attack the DNA to kill the cell. It's no longer something as simple, because if you go to the doctor and if you're not allergic to penicillin, you ask them, they'll give you penicillin, okay? Basically, if you're not allergic to it, go get the penicillin, it might cost you $2. Kind of interesting, isn't it? Just saying. So, now, if the pathogen can make it through all of this stuff that the body's going to give to it, all right, and we begin to enter into, maybe I've now got a fever, maybe I've got inflammation, maybe I've got pain. It might be that I'm going to have to get into a little more specificity. So when we have that exposure to a pathogen, well, is fever always a bad thing?
No.
No. Should you really, the minute it goes up 1 degree for your temperature, start taking Tylenol and Motrin?
No.
No. You should wait till about what temperature before you start that? About 100, 101. Okay? Other than that, leave your body alone. Because you know what your body's doing? It's fighting whatever that pathogen is. Now, you start getting over the 101, start getting a little concerned, okay? But if it comes down with the Tylenol or the Motrin, chances are it's okay. Now, fever is adaptive. It does promote interferon activity. That means fighting a virus, okay? It elevates metabolic rate, making more of the proteins of the cells, making more cells, so that we can get more interferons, so we can get more of the proteins, so we can fight more of the virus. But what do we want to do? We think it goes up to 99 and we should be taking something. This also is going to help with any type of tissue repair. Now, the temperature is also making your body not attractive to the pathogen. 98.6 was very attractive. That was the perfect temperature for that pathogen to exist in. But now it's like, whoa. I need to stop that. And temperature goes up. You have now made your body unattractive. And that pathogen doesn't like it, can't survive in it, and hopefully it'll die. Just saying.
So if you have, my son when he was little had febrile seizures, and they said when he got any temperature, they didn't care if it was 99, that I needed to give him. So is that something in that case?
That's a totally different.
Okay.
Yeah.
So this is just like if you're
this is just
sick,
just normal.
just like a basic flu, you feel yucky. Okay.
Yeah. Mm-hmm. Now, if there has been tissue damage, ta-da, think about that. Okay, that's the only place I think I have damage right there. And right there. Okay, so now, if you got some tissue damage, all right, usually there'll be some inflammation because of everything rushing to the site, okay? Now, this, once again, is local, it's general, all right? Body's beginning to attack it. We want to limit. The reason that we have this reaction is because we want to limit the spread of that pathogen. We want to try to destroy it, and we want to try to destroy it at the area of the breach. It wants to remove the debris of damaged tissue so that the healing can begin. Well, right there, all right? The signs, redness, swelling, heat, okay? May or may not have that depending on the extent of the breach. The processes. When this happens, we have now done mobilization of the body's defenses. They have recognized, whoa, there's been a breach. Let's go check it out. We want to contain and destroy the pathogens, and we want the tissues to get cleaned up and repaired. I don't know what happened to the rest of that. Did I miss that on y'all slides?
What are you talking about?
The repair, A-I, R. I didn't. Y'all write the R.
These are the three major processes of inflammation, correct?
Well, this is our body mounting the attack. So, when we mobilize the defenses, this is the most immediate requirement. This is really what the body needs to do very strongly. We're going to use that dilation of the blood vessels, which is termed hyperemia. because that is important in getting leukocytes to the area. Because if the dilation occurs, I can get those blood capillaries open and rush the blood to the area. When this happens, it is termed diapedesis. And this is a term that you will come across again. When that hyperemia, the rushing of the blood to the damaged site occurs, and those capillaries have dilated, and we're moving materials to the area of the breach, this is diapedesis. For the containment and the destruction, do you guys remember fibrinogen that's in the blood? That is going to get rushed to the area through the blood and begin to build the clot. Neutrophils, ta-da, just mentioned this, arrive to begin to start eating the bacteria. What did I just tell you? I just told you that neutrophils, okay, are one of the first cells to arrive. Their job is the same, eat the bacteria. Neutrophils will call up more macrophages. If we have to get specific, this can help promote production of more leukocytes in the bone marrow. For the cleanup and the repair, monocytes are a major player in the cleanup and the repair. They get there about 8 to 12 hours after the breach. They can act as an antigen-presenting cell, meaning if they're arriving 8 to 12 hours after the breach has occurred and there is still activity going on to fight pathogens, we now need help. We need to move to a different type of attack. This process of repair continues until the tissue is fully recovered. Now, if that monocyte has to act as an antigen-presenting cell, we now leave what is known as non-specific resistance, and we now have to get specific. So, we now have to move into adaptive immunity. This is my third line of defense. Everything up to this point was first and mostly second. But if 8 to 12 hours later, little monocyte comes along, recognizes this just isn't getting cleaned up properly, it will now become an antigen-presenting cell. This is the immune system. Everything up to this point was defense. I now have invoked, I have asked, I have incorporated, I have activated my immune system, which is a large population of widely distributed cells that'll recognize foreign substances and will act to neutralize and destroy them. There are two characteristics of my immune system. It is specific. It goes after one particular pathogen. Second, it creates a memory. Meaning, the second time, the third time, the fourth time, every other time, for just about the majority of your life, because it's not everything, okay? Even though there might be a memory created, it doesn't mean that it's going to protect you your entire life, okay? That's the goal, okay? But it does create memory so that all the next times that you're invaded or exposed to that same pathogen, it's going to take care of it. Good example. You got a cold last winter and you survived. You're still here. But this year, you get a cold again. If you had it last time, based on what I've told you so far, shouldn't you not get another cold? You should? Why do you get one?
Because there's 600 different kinds
Because
of them.
we've got so many different types.
Yeah.
You just
We'll get that thing.
got a different one this time. You got lucky, so to speak. Take a break, back at 9:30.
It makes me want to just catch them all.
Sorry, I just
They're like Pokemon.